हाँ कर दो।
Okay, sir. Thank you so much. Jai Hind, good evening and welcome back to all the respected dignitaries, distinguished speakers, faculty members, researchers, students and participants joining us from different parts of the country and across the globe. It gives me immense pleasure to welcome you all to this 10 days international training come workshop on advances and innovation in veterinary science for viable livestock practices. I, Dr. Nisha Rana, warmly welcome you all. This international training program is being organized by College of Veterinary Science and Animal Husbandry, Jalukie, Nagaland, under the esteemed Central Agriculture University, Imphal, in collaboration with Agricultural Forum for Technological Education of Farming Society and Vital Biotech Education Group. This program is being conducted under the visionary leadership of our patron, Dr. Anupam Mishra, Honorable Vice Chancellor, Central Agriculture University, Imphal, Chief Patron, Professor Indra, Director of Instruction, CAU, Imphal, Chairperson, Professor Lalita, Director of Research, CAU, Imphal, Convener, Dr. I. Shakuntala, Dean, College of Veterinary Science and Animal Husbandry, Dr. Amrit Gangoi, Convener, and Dr. Dinesh Kumar, the course coordinator, and Dr. Jitendra Mehta, President AFTEFS, along with the dedicated efforts of organizing committee and faculty members of College of Veterinary Science and Animal Husbandry, Jalukie. This 10 days international training camp workshop is being organized and conducted on an online platform Zoom, bringing together experts and participants to share their knowledge and innovations in veterinary science and livestock practices. So now moving forward to the technological session. For technical session first of day 5, I would like to introduce our first speaker, Dr. Vidya Singh, the senior scientist and plant pathologist, ICR IVRI, Izzatnagar. So, Dr. Vidya Singh is a senior scientist at ICR IVRI Izzatnagar. He did his graduation in veterinary science from Madras Veterinary College, TANUVAS. He did his master's and PhD in veterinary pathology from IVRI Izzatnagar. He joined ARS services in 2009 and served as National Research Center on Mithun in Nagaland for 6 years. He got transferred to IVRI in 2016 and since then he has joined working on cattle disease to establish the occurrence of different pathogens in country. He has handled many externally funded projects from DBT, DST, RKVY, ICR network projects as principal investigator. He is currently working in the area of use of computational biology and immunoinformatics for the development of next generation diagnostics and vaccines of the respiratory and enteric pathogens. Today, he will be discussing about the computational biology and its application in the development of next generation diagnostics. So I would like to request to Professor Dr. Vidya Singh to kindly deliver his keynote lecture for technical session first.
Thank you, Dr. Nisa. Am I audible?
Yes, sir, you are
Audible, sir.
Good. So, good evening, everyone. Thank you for inviting me for this lecture on the computational biology and its application in the development of chimeric antigen-based diagnostics. Computational biology, the name itself indicates the use of some computation, use of computer and computation. Computation means ganana, some calculations. Or this computational biology is not new thing. It's not new thing. It has been there in the existence in different civilizations over hundreds and thousands of years, where just by observing the patterns of the stars and other things, things were being predicted when the solar eclipse will come, when the moon eclipse will come, and how many days it takes by the moon to take one rotation around the earth, or how many days it takes the earth to take one rotation around the sun, and like this, so many. So based on these patterns, many many scientists or this forefathers, they have depicted, okay, this is the particular pattern by after which the sun comes to this particular area or moon comes to this particular area, and like this they have depicted, they have recorded. Similarly, in recent in past few decades, we have used these calculations, we have used the identification of different types of patterns in molecular biology, in disease diagnosis, and many other fields where we have tried to identify different patterns. Okay, suppose if this particular sequence of amino acid is there, of 10 to 15 or 20 amino acids is there, if this particular sequence of amino acid is there, this particular sequence will be responsible for providing allergenicity to a particular protein. So similarly, many different sequences of amino acids are there. They have been pointed out. They have pointed out that if this particular sequence is present in any protein, that protein will serve as an allergen. Similarly, if particular sequence of amino acids and nucleotides is there, that particular nucleotides or amino acid sequence represents toxicity. So if that particular sequence is present somewhere, that particular protein will be have as toxin. Similarly, if particular sequence is there, if that particular sequence is there, that particular sequence can behave as an immunogen, that can behave as an allergen, that can behave as an antigen, and so many like this. So using certain things, using these patterns, now there are many databases are there. Close this now. There are many databases are there for peptides, for proteins, for allergens, for toxins, and many other and those sequences which are which have got the potential to be called as immunogen, which have got the potential to be called as antigenic epitopes and all. So there are many things. There are certain sequences of certain biomolecules. It can be certain organic or inorganic sequences. If they are responsible for acting as a drug. If a particular sequence is there, that can bind to a particular receptor. If particular sequence is there, that can work, that can stimulate our host immune system to produce antibodies against that, to produce vaccines, to produce this antibodies against that, and to produce cell-mediated immunity. So these
Excuse me,
sequences.
sir.
Hello.
Sir, excuse me, sir. Slides are not moving.
No, I'm just not moving. I'm just giving instruction. I will be moving it now. So I will be discussing here in first few slides about the theoretical background. Okay, what is computational biology and what are the different types, what are their components and all. And then
Okay, sir.
subsequently, I will be discussing about my own work, which I am carrying out in my own lab using this computational biology for the development of this chimeric antigen-based diagnostic. So here you can see that computational biology is interdisciplinary science. Sorry. It is an interdisciplinary field which integrates biology, computer science, mathematics, statistics, artificial intelligence, and systems biology. It uses many computational and analytical tools like many algorithms, many mathematical models are there, many statistical methods are there to study any given biological system. And different types of biological data, complex from simple to complex biological data at different levels, at molecular level, at biochemical level, at cellular level, at organism level, at mass population level, that can be analyzed using this computational biology. What is the scope of computational biology? As we discussed, that large number of data are there. Large number of data have been generated or have been generated over past few decades. So how to handle this large many data, so large scale of biological data sets are there. So how to handle this? It is very difficult. It is almost impossible to analyze all these data, to store all these data at a particular place and interpret it. So using this computational biology, if certain data are being fed, just like our computer is having, suppose, lot many information. Many times we are not able to find out where is that particular information is there. So just we used to find using certain shortcut keys, Control F, and we used to type that particular file name, which may not be the accurate file name, but somewhat similar. Then our computer will give, okay, these are the possible documents which are having similar words in this, and then from there we used to find it out. Similarly, the computational biology, if many sequences are there and we are trying to find some known or unknown sequences, if you just will find it out, they will give many interpretations. If some unknown molecule is there, they can give similar molecules which have been submitted in the database, and based on that they can give their predictive function, predictive structure, all these things and all. It improves accuracy. Why to use computational biology? Because if if these things can be done, just suppose you in our computer so much of data is there, and if I don't remember that particular file or that particular document where it is saved, if that control if option would not have been there, then it may take even hours sometimes for us to find that particular file. Similarly, in computational biology, that when by using this computational biology, that speed, accuracy, and predictive power that improves tremendously. It expands our research from lab-based to data-driven biology. Whatever work we are doing in the lab, that is being recorded in the form of data, and that is being analyzed by ourselves or other workers across the world. It enhances the efficiency of microbiology tools. It has got wide range of applications in sequence analysis, omics technology like genomics, proteomics, and all transcriptomics. In a structural biology, in the study of protein, RNA, their modeling, structure, predictions, systems biology, networking modeling, to find the pathways of different molecules, to find pathways of different pathogens, how they are entering into the cell and how they are reacting, which part of the cytoplasm or nucleus, and what changes they are bringing about, what types of cytokines they are bringing about. So that is a network modeling. Machine learning, we are training our machine. We are training machine by different. We are training the, suppose one example we will give. Suppose that particular one bone is there. So we can train our software, we can train our machine by giving hundreds of and thousands of pictures of a particular bone. And then after that, if once the machine is trained for that, then by just taking the picture and submitting into it, that software, that machine will give, okay, this is humerus, this is femur, this is radius, this is tibia, this is ulna, like that. So it has got applications in development of new drugs and vaccines designs, epidemiology and disease modeling. As we discussed, that it has got application in multi-omics technology like genomics, which involves the analysis of DNA sequences, large number of DNA sequences, starting from the small sequence of Sanger sequencing results to NGS sequences, which comes in gigabytes of the volume. Transcriptomics in the expression of RNA, then proteomics, protein profiling and function, metabolomics, as we discussed, that different metabolites and their pathways within the cell and outside the cell and all. Integration of multi-omics, it helps at system level understanding. What is significance? As we discussed, that computational biology, it has transformed tremendously the traditional biology into predictive science, using the works carried out by different workers across the world, and which has been submitted into the data. Then we can analyze it, and then we can do predictive. Okay, this type of work it has been carried out, so what else can be carried out, and which can be predicted. It facilitates generation of research hypotheses. It improves experimental design and analysis. It reduces the dependency. Earlier, what was the trend? That okay, this particular drug is there, we will just administer into some rats or mice or other laboratory animals, and we used to study, okay, what is the toxicity study over a period of time. But now, gone are the days. We will just take that particular molecule, whether it is known or unknown, we will just take its sequences, we will just take its structure, and we will submit that particular sequence or structure into the database. And the database will give, okay, this particular sequence or this particular molecule, organic and inorganic molecule, which has been submitted, that is somewhat similar to molecule A, to molecule X, or to molecule A123, like that. And if suppose out of 10,000 or out of thousands of molecules which have been submitted to the database, this particular our test molecule is resembling with 4 or 5, that will give one this, okay, that will screen, okay, this its a structure is 99% similar to particular type like A or B or C or D, whatever it is. Then once the structure is similar, so the function may also be similar. So like this, we used to screen down, and that has increased our this one. So it reduces the dependency on trial and error. So hundreds and thousands of trial and errors can be reduced to carrying out only 5 or 6 or 10 trials and errors. Suppose out of 1,000 molecules, we have screened down to 5 molecules. So we need to carry out trials for only 5 or 6 molecules, like that. So it enables handling of high throughput data. It enables rapid pathogen detection, it enables development of fast diagnostic and therapeutic design. It enables personalized medicine. There are certain persons which will be allergic for a particular molecule, which will be who will be allergic for particular organic or inorganic molecules. So for those particular persons, the personalized medicines can be done. Okay, this particular person is allergic, so against them particular other medicines which will be non-allergic to that particular individual, that can be designed and done. So what are the key drivers? What is the need of computational biology? As I told that there's so much of in last few decades, so much of data has come out of various types of research are being carried out, just like sequencing data. Just imagine in in early 2000, where when the first human genome was this deciphered, only one sequence was there, and then in last 20-22 years, the number of sequences that has gone to tens of thousands. So imagine NGS sequences for a particular that single human being, that itself is very difficult to analyze, and now tens of thousands of these sequences are available for different species, for different species, and again in the same species for thousands of persons it is available. So how it can be realized? So something was required, something was required, some computational method was required by which all these data can be realized. So this big data and NGS revolution that has led to the development of big data and NGS revolutions. Development of AI and deep learning. What I was telling that we can train our machines, we can train the softwares for different suppose for different diseases that we can train. Okay, if temperature is there like this, if then these are the clinical signs, these are the these gross lesions, these are the microscopic lesions, these are the specific points. Like suppose if we have trained that in particular cell, if intranuclear inclusion body is there and that is present along with the nervous signs, then it may indicate, okay, nervous signs, there are certain diseases which shows nervous signs in dogs, and in that case the microscopic lesion includes the presence of intracytoplasmic inclusion bodies, that is negative bodies, that we can get diagnosis. Okay, it may be a case of rabies, like that. So like this, it can be done wherever it can be done where experts are not available. Also availability of the cloud computing platforms and emergence of systems biology approaches that has led to the development of computational biology concept. And we can see the components of computational biology. There are bioinformatics, which deals with the alignment of sequences of DNA, RNA, proteins, and all, genome assembly, functional annotations and function profiling, homology search. Just I was saying that if particular sequence is there, so we used to put into some database like BLAST and Hammer and all, and that used to give, okay, this particular sequence is matching 50% or 60% or 90% with the particular sequence of this species or this bacteria or this virus and all. So detection of variants because of the mutations and SNPs, phylogenomics. Then computational genomics also help us to find out genome-wide association of sequences, genome-wide patterns and variations. What are the different patterns are there across the genome? Then comparative genomics, that helps us to find the evolutionary relationship between different strains or different bacteria or different viruses or different individuals or different species. And pangenomic studies that help us to find the population diversity. Coming to the AI and machine learning, that all we have discussed, that it helps us, if a software is properly trained, that will help us to find out to predict the disease. It helps in AI driven drug discovery, machine learning, diagnostics, and biomarker identification in a particular disease. Okay, suppose if you are training machines for a particular disease, suppose disease is or rabies is there, we can take the blood samples and all possible samples, and the results of that we can enter into the database. Like this, thousands of persons, this data that will be entered into the database. Then after once the data is entered for tens and thousands of persons, then that machine will find, okay, all the rabies positive persons, they have got this particular biomolecule increased level, this particular biomolecule has got significantly decreased. Like this, we can go for identification of certain biomarkers. And similarly, this systems biology, like this analysis of or a study of or exploration of the different pathways, metabolic pathways of the viruses, bacteria, cytokines, different molecules within the body that can be explored. The interactions of protein with other proteins and receptors and other host pathogen interactions, all these things can be carried out under systems biology. The exploration that another component is there, structural bioinformatics, which deals with the three-dimensional structure of the proteins, which can be done by using different bioinformatics tools like AlphaFold, Rosetta, and all. RNA structure modeling, molecular docking. Docking is like that. Okay, if one protein is there, what unknown protein we have got from somewhere. Suppose we have taken some samples from some area where a human being has not reached. It may be Antarctica, it may be Mars, it may be Moon, or it may be some other planet, and we have got. We have taken out and we have extracted the protein, and we have sequenced that protein, and then we are submitting that particular sequence into the protein database, which is having more than 40,000 proteins. This data is there. So based on the sequence of this particular protein which we have extracted from the soil or material which we have brought from the Mars or anywhere else, that machine will give us, okay, this particular molecule, unknown molecule, is 50% similar to bacteria this or virus this or this particular species, like that. So based on this, that we can find out, okay, if particular this it was there, so some organism was present there which was having, suppose that particular protein what we have got that is having function similar to insulin, based on our this computational biology, then we can predict, okay, some organism would have been there in that particular planet which was using insulin for some or the other this metabolic activity in its body. And like this, and another function is molecular dynamic simulations. Earlier, what we used to do, that for finding out, okay, this particular molecule, it can enhance the immune response, it can stimulate the immune response or not of any animal. We used to give that particular molecule as injection into laboratory animals. Then we used to collect blood, serum, and other samples over a period of days. It may be for three weeks or 4 weeks or 5 weeks or 10 weeks, like that. And then we used to analyze in our lab. But now there are softwares, there are machine has been trained wherein that particular sequence, if we'll be submitting it in that machine, in that software, there are certain things. Those functions, there are certain nodes which will function as our lymph node. There are certain nodes which will be functioning as the antigen presenting cells, like dendritic cells and macrophages and all. So they will analyze, they will react with these sequences, and then they'll give, okay, after once this particular molecule is injected, so this particular cytokine can be increased by this many times. This particular cytokine will be its expression will decrease by this many times. All these things can be this predicted. It can be simulated like this. So like this, what we can say that these are neural network of the biological data input and diagnostic output that we can take. We can see at one side that patient samples and data, like genomics data. It can be there based. It may be DNA sequencing, RNA sequencing. It can be proteomics. The protein what we have got, unknown protein is there. We can analyze this sequence by mass spectrometry or any other things. Metabolomics, metabolic profile that we can see, and the clinical data and all. All these are taken as input, and then by AI or deep learning or machine learning, all these hidden layers of feature learning are in the different interactions, and this happens, and that all these different interactions give us 2 or 3 or few more diagnostic outputs. It may be diagnosis, it may be A or it may be B, and then based on that, again we can find it out the prognosis and other things, that okay, we now we are restricted, okay, we have to differentially diagnose this particular these two diseases only, A and B, like this. So the computational biology, now coming into our areas which we will be discussing in detail and in which we are working. We are working for the use of computational biology. We are using different tools of computational biology for the development of diagnostics. This computational biology integrates high throughput sequencing, NGS, bioinformatics pipelines, artificial intelligence, machine learning models and all, and systems biology. It enables faster diagnosis, high accuracy, and personalized dedication and treatment. One among the things is here, next generation sequencing, as all of us knows what is the importance of next generation diagnostics. Next generation diagnostics is there that it should take minimum time. It should take minimum time. A large number of samples can be analyzed. It should have high resolution. It should give precise diagnosis. It should be given, and any pathogens should be diagnosed at its early stage so that it will help for its therapeutic and prophylactic management and all. And then personalized host response can be it is possible using next generation diagnostics. The advanced computational tools like that we're discussing, the different software, different machines are there, different pattern recognition. What I was talking just before the start of our talk, that pattern recognition has been there in the existence in different civilizations across the world. Okay, this particular pattern is there, it means summer will come now. Then this particular pattern is there, so now night will come. If this particular pattern is there, so after a few days solar eclipse will come. Like this pattern recognitions. So we have trained, and we are training also the scientists across the world, the neural network, just like the networks in our brain. So like this, different networks are being made which can recognize different patterns, and based on these patterns analysis can be given. Graph-based models and cloud-based bioinformatics pipelines like big data handling and probabilistic frameworks based on, okay, this data, this is the probability, like that this can be given. And so it has got wide range of applications in the medicine, in the detection of cancer, in the detection of genetic disorders, in monitoring of antimicrobial resistance, and it enables this discovery of novel pathogens. As I was discussing, that known and unknown pathogens, the sample which has been brought from somewhere else where human civilization has not reached, and some unknown pathogen is that also can be analyzed, and that can be analyzed without the risk of handling it, without the risk of getting infected. Why? Suppose some pathogen is there, if you have some virus is there, if it is infectious, so we immediately just we can take it and we can put into some samples which will neutralize it. Suppose, for example, formalin and many other chemicals are there which will kill the live virus, which will kill the live bacteria. But the bacteria or pathogen that is killed, but still its genetic sequences can be analyzed by using different molecular biology tools like PCR, real-time PCR, and whole genome sequencing. And then this sequence can be compared with the sequences available in the database. Then you can find, okay, this particular sequence of this particular unknown microbe is not matching with any of the microbes. Or something new is there, or it is matching only the 10%, something like that. So that novel pathogen discovery is being done like that. Real-time biosurveillance is possible. It supports pandemic preparedness. Just recently, as during COVID, use of this big data and computational biology that helped to counter this preparedness. Therefore, that led to the development of a vaccine within 1 year, which normally it used to take more than 10 years for any particular vaccine to be developed and to come to the field level. As we discussed, that we can identify that different biomarkers. Suppose there are hundreds and thousands of persons suffering from particular disease. We can take many samples. We can take all the data, all many investigations. We can do it, and then we can feed our machine. And from there, they will be finding out, okay, in thousands of persons, when this particular disease A it was there, so this biomarker it was increased at early age. This biomarker was increased or decreased at late age or mid age, like that. So these are called as pathogen signatures, or host biomarkers can be identified. The different softwares, different tools are there which has been prepared, like DESSEC, SR, Random Forest, SVM, many are there. Like, then another important tools for computational biology, like CRISPR-based diagnostic. It's advanced genome editing tool. It's highly specific. It targets gene. A particular gene can be deleted. Suppose one pathogen is there where 10 genes are there, and out of which one particular gene, like suppose gene C, that was responsible for infectiousness, or that was responsible for pathogenicity. Gene C was responsible for pathogenicity, and gene D was responsible for immunogenicity. So what we can do by using this technique, that we can delete that particular gene C, which is responsible for creating lesions, which is responsible for pathogenicity. So the gene C will be deleted, so that the gene deleted pathogen that will not be able to produce pathological lesions to cause death. But since other genes are there, like gene D is there, which was responsible for producing immunogenicity, so antibody will be produced against that. Antibody will be produced, and animal will or animal organism will remain this seropositive for that. So there are different patterns like Serlock, detector, and fellow. That different patterns are different platforms are there where this CRISPR based diagnostic is being. Out of it is Feluda. This platform has been developed. This is an indigenous. It has been developed in our country itself. So coming to the main concept, how I am using this computational biology and bioinformatics tools for the development of diagnostics. There is one term like multiple antigenic construct. Just I will start with an example. Suppose in a class, suppose class 8, there are in a school there are 4 sections of class 8: section A, B, C, and D. And in all the sections there are 50 students. There are 50 students. So out of 50 students, 50 students in a class, all the students will not be having the same level of IQ. All the students, they will not be having same level of understanding, or they will not be having same level of responsiveness. Some students, they will be intelligent, they will be having IQ, they can understand quickly, they can respond quickly. So another section is being made by carving out top 10 persons from all these 4 classes, and section E is being created. So what will be the result of the section E? The result of section E will be much more higher as compared to the depreciated section A, B, C, and D. So same thing, same principle is there in our proteins also. Whenever any infectious, any protein, suppose any virus is entering into our body, what is happening with that virus, or any bacteria, any pathogen, any antigen, say any antigen, what is happening? That particular antigen is being carried to the local lymph node. It is being carried to the local lymph node, and there within the skin itself, that is being processed by antigen presenting cells, dendritic cells, macrophages, and all. And that particular antigen within the macrophage or antigen presenting cells, that is being broken down into multiple fragments, just like this. Any antigen resembles a train having 30, 40, or 50 bogies. So each bogies, there are different bogies. So within the macrophages or antigen presenting cells, all these bogies are separated. They are cut. Between the bogies, certain sequences are there which are linker sequences. So these bogies are cut, and then similarly all antigens within the macrophages, they are cut into a small fragments, and each fragment is called an epitope. Each fragment is called an epitope. Now these epitopes are they bind with MSC molecules, and then that molecule, this MSC molecule bound with that epitope is being carried out to the surface of the MSC molecules, which present it to the T cells, CD4 cells. Now these CD4 cells that stimulate the B cells or T cells to form plasma cells or CD8 cells like that. So if plasma cells are formed, they will lead to the initiation of HMI, humoral mediated immune response. And if this T cells are stimulated, they will lead to if CD8 cells are stimulated, they will lead to the generation of cell mediated immune response. Similarly, and out of this all these bogies, there will be certain epitopes which are having more immunogenic, which are having more immunogenic or antigenic. So what we used to do using the bioinformatics software, we used to identify, okay, out of suppose 40 epitopes are there. So out of these 40 epitopes, how many epitopes are there which are present on the surface, which are immunogenic, which are antigenic? And we used to identify those epitopes, and we used to make a section E. Just like we made a section E of the top 10 students. So we used to make a different we used to make a different gene, chimeric gene, or multiple antigenic construct. And these all epitopes, they are this joined with certain linker sequences. Linker sequences, just like between two bogies, some connection is there. So just like linker sequences are being this design, and it is called a multiple antigenic, multiple epitopic construct, or multiple antigenic construct. These constructs are designed so they will be having highly immunodominant, immunogenic, and we used to find, okay, all these epitopes should not be none of these epitopes should be toxic, none of these epitopes should be allergic, none of these epitopes should have any negative characteristics for an immunogen. And now this MAC, this multiple epitopic construct, will behave as a section E of our class that will have the potential to stimulate our immune system to produce large amount of antibodies or a stronger CMI response. What else we can include in this construct? We can include certain adjuvant sequences. we can find out, okay, out of these epitopes, certain epitopes should be, certain sequences should be there which can induce our interferon gamma, or IL-4 or IL-10, which are required for our HMI and CMI responses. And what I was talking just now, this is a technique wherein we need not use highly this hazardous pathogens like live viruses. There is no biosafety issue, no risk of latency, and has got DIVA capability also. Because suppose COVID itself is there, I can for working with the COVID virus, we need to have BSL 4 plus level facilities. But if you are working with this, just we have to take the sequences which is available in the NCBI. We have to compare the sequences submitted across the globe, and then we have to analyze the conserved sequences. Then we have to find out, we have to design the construct, we have to get it synthesized, and then we can clone it, express it, and we can work for the development of diagnostics as well as vaccines. There is no need of any biosafety level. Since we are not handling the live viruses, we are just taking the sequences which someone has submitted. Or suppose if live virus has come to us, then also just we can, at the time of receiving itself, we can put certain chemicals which will kill the virus. It may be formalin, it may be another, and then that dead virus is there, that can be processed and its sequence can be analyzed by NGS or another sequencing methods, and then this particular sequence can be used to find out these epitopes which has got the potential for enhancing this B cell epitopes or anti-cell epitopes for HMI and CMI and all. And it is being carried out across the world for many different pathogens. So many different diagnostics and vaccines or therapeutics are under different stages of clinical trial, like this for human monkeypox viruses, for human enteroviruses, swine hepatitis E virus, H1N1, CCHF, chikungunya, HIV, mycoplasma pneumoniae, BSE, scrapie, mycobacterial infections, BTV, blue tongue this virus, and toxoplasma gondii, and many more. Many more are being carried out. And to add to this list, we ourselves in our lab, we too are in the process of doing for many diseases like BPI3, BRSB, rotaviruses, coronaviruses, and all that we'll be discussing again when we are showing our own work here. So, as I was discussing that we have to find out different antigens, different epitopes, and then in between the different epitopes, we have to put some linker sequences. We have to put some linker sequences, and then cleavable sequences, adjuvant we can put as we can see on the left side, and then on the right side that we can put certain tags, histidine tags or flag tags and all to monitor our this molecule, and then targeting moiety. We can put certain sequence of our antibody also, which will guide this molecule to get to bind easily with the antigen presenting cells and other cells and all. Here, one more thing is there, like this. What we can say, one single construct can be prepared. One single construct can be prepared for one pathogen, two pathogen, or more pathogens. Suppose one or two. Suppose we are working on respiratory pathogens. So for respiratory pathogens, there are some common viruses are like PI3, BRSB, then coronavirus, herpes virus, BBD. So we can take few epitopes from all these 5 pathogens. Even some bacteria also, like it can be Pasteurella multiflora, it can be Klebsiella pneumonia. We can take suppose 10 epitopes from all these pathogens. We can form a single construct, and the protein expressed from this, that can be used for immunization of the animal, which will produce its HMI, which will enable the host to produce antibodies against all these pathogens. Once antibody is there, so any one of these pathogens, if it is being introduced into the host, then that will get neutralized, and the progression of disease will not be there. So the different antigens can be incorporated either from the same or different strains. Suppose many times what is being done, normally any diagnostic vaccines traditionally, what they used to do, they used to isolate some bacteria, they used to isolate some virus, and then from that, suppose in one lab 10 or 100 isolates are there, so they will take one particular isolate, and against that isolate they will be making vaccine by killing it or by inactivating it, or by using one isolate. They used to go for development of diagnostics. But any diagnostics, any vaccines based on a single strain will never be 100% effective. Why? Because there are, suppose E. coli itself is there, so more than 200 these strains are there. If you are taking one strain, so one strain does not match completely with other strains. So in that case, what we used to do, What we should suppose whatever hundreds of suppose 100 strains of E. coli is submitted there in the NCBI. We should take the sequences of all the strains, then make phylogenetic analysis. Then suppose 10 clades are there. So from each clade, one prototype strain has to be identified, and then from each prototype strain, 2 3 epitopes will be selected, and then all these 2 epitopes, it will combine to 20 epitopes. So if multiple antigen construct is prepared by taking 2 epitopes from each clade of the phylogenetic tree, then the antibody or protein which will be produced here, that will be reacting with all the strains of that particular clade. So like this, we can combine and we can pick a universal universal chimeric gene which can be used for immunization of a particular pathogen, which can be used for the development of a diagnostic for that particular pathogen of all the strains. So here, what we can see, just for example, just here we have taken an example of our this bovine herpes virus 1. So there are these are the genes GB, GC, GD, glycoprotein B, glycoprotein C, D, G, H, I, E, and these are present on the surface of the virus. So that we can see here in the graph on the right side, that we can see whatever yellow peaks are there, they represent those epitopes which are immunogenic, which are antigenic, and that's why they are projecting upward positively from the baseline. And those peaks, those and those which are green in color, and then they are projecting negatively or downwards from the baseline, they are non-immunogenic, they are non-antigenic. Okay, so what we have done, suppose just you can see here, we have selected these yellow peaks, those epitopes, those nucleotide sequences or amino acid sequences which represents this yellow peak from glycoprotein B, C, D, G, H, I, E, and all, and we have make one multiple antigenic construct which you can see as MAC. Here we can see more than 80% of the sequence that is having yellow color peaks. So the intensity, you can see it here, intensity of immunogenic epitopes, intensity of this antigenic epitopes is much more in this. Though the sequence length is same, it will be similar. And when this MAC, this construct will be used for as an immunogen, as a vaccine candidate, that will have a much better effect as compared to normally what people used to do. They used to express one particular gene, one GB, a GC, a GD, and then they will use that this express protein, recombinant protein, to evaluate its diagnostic potential or vaccine potential. So any one gene, any one single gene will be having very much less potential then this MAC to be used as diagnostic or vaccine. So here, how we used to process? Just we used to retrieve the sequences of the surface protein from the NCBI or other databases, PDB and all, and then we used to predict the linear B cell epitopes and T cell epitopes. So there are different softwares like immunogenicity. One particular character is there, characteristics that has to be predicted with IEDB, and many other softwares are there. Just for example, one software for each I have noted down. Like for immunogenicity, we have to put that sequences into IEDB. Antigenicity, we have to put IEDB AR. Allergenicity, allertop, allergen, toxicity, toxin parade, and whether this particular sequence or epitope that is having transmembrane topology. So that TMH is there. Then conservancy has to be analyzed. That particular selected epitopes that should be conserved across all strains. That is epitope conservancy analysis tool. Resemblance to host protein. If any particular epitope we have selected and that is cross-reacting with the host protein itself to the tune of 50, 60, or 70%, then if that epitope will be used, then that protein will act as allergen and that particular individual will develop allergic reactions. So all those sequences, all those epitopes which cross-react with the host protein, they are to be discarded before constructing this multiple antigenic construct. So all cross-reactive epitopes has to be eliminated. Then all these selected epitopes, B cell epitopes, T cell epitopes, has to be linked with the help of certain flexible linkers and rigid linkers, depending on our this purpose. Then once this MAC is constructed, again it has to be analyzed. Again it has to be as a whole construct, and that has to be analyzed. Now whether it is immunogenic. Single epitope was immunogenic. 10 epitopes we have selected. All these 10 epitopes individually they were immunogenic, but as a whole combination, whether they are immunogenic or not, whether they are antigenic or not, all the physicochemical properties has to be evaluated using softwares like Peren, Ispaxi, molecular weight, this isoelectric pH, then amino acid composition, extinction coefficient, instability, aliphatic index. All these things it has to be estimated, and wherever some protein or some component is failing, that we have to discard it. Then we have to go for prediction of secondary structure using software like PDB sum, and go for then protein structure modeling, refinement, 3D structure, Galaxy TVM, Galaxy Wave, iTeaser. These are the softwares which help us to find out the protein structure modeling and refinement. Protein structure is refined using Galaxy Refine, and solubility, whether that particular protein will be soluble or not. For that, again, this Sol Pro software is there. And then once it is passing all these things, then we can go for generation of PDB file, that PDB sum, and then that PDB file has to be further validated for its structure and 3D model using different software like VADAR, IRAC, ProChex and all, and then selection of the base model. Base model has been on the right side that we can see there at Ramachandran plot analysis. We used to do this analysis to find, okay, most of the residues, more than 97-98% of the residues should fall into the most favored and favored regions. Okay, and then we can see here, suppose for each final model, for each structure sequence, the software will give 5 or more models. Out of these, the best model has to be selected based on their negative energy, the delta G value and software. And once we have finalized, okay, this particular model is there, then we used to take that sequence, and then that particular sequence, now it depends where we are going to express that sequence. So that sequence has, if we are targeting prokaryotic vectors, eukaryotic vectors, yeast, or this bacterial virus, then accordingly we have to optimize the codon. Suppose what we are doing it here, in prokaryotic vectors. So using JCAT software, we will optimize the codon, and once the optimized sequence of nucleotide that we are giving to this commercial companies for synthesizing it, they used to synthesize and giving certain vectors. It may be PATE, it may be PUC, or any other vectors they can give it. And once we are receiving it, then we are going for the molecular biology works, like we used to amplify them, we used to clone them, we used to transform them, and then we used to go for this induction of expression of protein. And the protein express is analyzed by SDS phase analysis, that we can see it here, that this picture depicts for two particular proteins. One is here, which is having 42 kDa, and another one is there, which is having 29 kDa. So 29 kDa is or this PI3 virus, and 42 kDa is BRSA virus. So these two proteins were the first two proteins which I designed, the chimeric genes which I designed 2017, and then we carried out the work. And that we can see that how much thick band is there. This chimeric gene has led to the production of very thick band of this chimeric peptides. And then here we can see there that Western blotting analysis using anti-histag antibody, that we can see very thick band is there, that we can see. And then these proteins were inoculated into the laboratory animals like rat, rabbit, chickens and all, and then the serum collected from these animals were were subject to carry out this blot analysis, that we can see here. It reacted. This pre-sera, there is no reactions. Before immunization and after immunization, that protein has reacted. That has led to the thick band here. Then again here you can see here, and the pre-sera there is no band, and post-sera band is there. Similarly in the rat and the chicken also we did. Again the dot blot also you can see that we have done it. For VRS we had the negative sera and the positive sera. Now what we did, once we analyzed, okay, this protein is reactive and it is producing very good immune response, and it is producing a very good amount of these antibodies in the serum, which is reacting, as we have seen in there, by showing a very thick band of this. Then we further purified this protein. By going HPLC and size spectrum chromatography, and we took this particular fraction, this particular single P, which represents only that our target protein, and then we have gone for optimization of this diagnostic like ELISA. So that we have gone for. Now the checkerboard titration, that we can see it here, that the protein was so pure, and thick, that when we carried out checkerboard titration, they're using non-positive and non-negative sera samples, that we can see in checkerboard titration. This is the picture what we got for both the viruses individually. This is the picture of only one virus, so that we can see that check out the bookish picture, that it ideally it should come, that concentration, the color should decrease downward, and it should decrease this horizontally also, and same thing which has occurred. And based on this, here what is that, and we have taken different dilutions of antigen here in A, B, C, D, and this vertically, and horizontally we have taken different dilutions of the positive serum up to 5, and then different dilutions of negative serum from 7 to 11 onwards. Then we have and then based on the results of this, and again we found out that okay, as per the convention, there's by P by N ratio, it should be more than 5. It should be more than 5. And then we finalize even that at 16 nanogram protein level, that was producing a very good results. Here we can see that the ratio P by N ratio, even at 8 nanogram also it was being produced. But for being on the safer side, that we took it at this 16 nanogram, and the serum dilution was taken as 1:25. Then using this 1:25 serum dilutions and 16 nanogram protein, we checked known negative samples from the commercial from the commercial antibody ELISA kit, and we found it out that the cutoff value of it was 0.18, which was much lower as compared to the commercial ELISA kit also. So because commercial ELISA kit, that was based on the recombinant protein of only one particular protein, but here we have used the epitopes from 3 different genes which are present on the surface of the virus. That's why it has increased the sensitivity and specificity of this particular protein. Here just a look of other pathogens that we discussed, like PI3, BRSB, SRLB, 1 coronavirus and BBDB, that we are working on. We have prepared multiple and constructed, we have cloned, expressed, and we are in different stages of development of this diagnostics. So what are the advantages of computational biology? The computational biology is revolutionizing the diagnosis. It enables rapid, accurate, and personalized solutions. It integrates biology with advanced computing. It is very critical for future healthcare, agriculture, and biosurveillance. And it is what is the future? It is the computational biology is the future in every field, whether it is diagnostics, whether it is therapeutics, whether it is vaccines, or any other area, because it makes this real-time biosurveillance, real-time disease monitoring, and clinical decision support is possible using computational biology. It helps in AI-driven healthcare and intelligent diagnostic systems, precision veterinary, and human medicines. So what is there in front of us is just an iceberg. What the potential of the computational biology, AI, machine learning, these things and all, and much more is still hidden into the ocean, which needs to be explored. So it's all from my side. If anybody is having some queries, they can ask. I will be happy to settle their queries.
Thank you so much, sir, for enlightening us. So participants are requested to kindly post their question in the chat box.
Thank you, sir, for wonderful presentation.
Thank you. Thank you. Well, one thing I would like to tell, actually, there are many bioinformatics institutes. There are many bioinformatics institutes wherein this dry lab work is being carried out. This bioinformatics study is being carried out. Epitopes are being identified. These potential drugs, these molecules are being identified. But there is no collaboration of this bioinformatics institute with research labs. Research labs where wet lab is being carried out. But in our lab, what we are doing we are doing both. We are doing dry lab by using the analysis of the sequences, constructing our own construct, and analyzing those constructs and proving those constructs whether they are working or not by carrying out wet lab research. So it's a and in India, very few institutes are there wherein such works are being carried out. It will be very so much of potential is there. So if some of you should undertake research work in this area, and what I told that I did not have any formal training. I did not have any formal education in bioinformatics and computational biology and all. I underwent this computational biology training just by reading articles, by seeing some videos, and just listening, and over a period of time. And then I developed some expertise wherein I am just doing these things. So all of you can move in this area also, which has got tremendous potential.
Thank you so much, sir, for such an informative session. We are very, very thankful to you.
So okay, thank you.
So moving forward for the next technical session too. For next technical session
तो हम भी करते हैं
too,
सर, लेकिन यहाँ कंट्रोलर मैडम ने
I would like to introduce
फाइल में लिख
participants
दिया
are
था, इसलिए जो है ना
requested to
सर डिले
kindly
हो गया,
mute themselves.
अदरवाइज मैं कर सकता था सर। नहीं,
Please
मुझे
mute.
पता नहीं था सर, कंट्रोलर मैडम ने डायरेक्टर सर को लिख दिया ना सर, मार्क कर दिया।
Mute your
और नहीं एक्चुअली उन्होंने इसलिए मार्क कर दिया होगा कि शुरू में जो है ना ये 18 लाख के लिए अप्रूव था, बाद में हमने फंडिंग से इसको रीअप्रोप्रिएट करवाया 26 लाख 90 हजार के लिए। हाँ ठीक है, मैंने
डन कर दिया।
ठीक है सर, हाँ सर,
दे कैन अनम्यूट योरसेल्फ।
हाँ सर, अप्रूवल करा लीजिए सर, सैंक्शन मैं कर दूंगा सर। ठीक।
So moving forward to the next technical session.
सर,
on behalf of organizing committee, I would like to
ठीक है सर,
participants,
नहीं ठीक है
all
सर,
the
नहीं मैं ही
participants, and
करता सर,
also thank
कंट्रोल
you to
मैडम
the
ने
speaker
डायरेक्टर को
one
मार्क
for
कर दिया
technical session
और
one.
मैडम चली गई।
So I'm forwarding to the technical session 2. So for technical session 2, I would like to introduce our second speaker, Dr. S. Kumar Das. Sir is Associate Professor in Department of Veterinary and Animal Husbandry Extension, College of Veterinary Science and Animal Husbandry, Central Agriculture University, Imphal, Jalukie, Nagaland. So sir is also a renowned scientist, and sir has published many research papers in many reputed publications at national and international level. So now I would like to request you, sir, to please deliver your lecture note.
Just a minute, sir. Sir is joining.
Okay. So participants are also requested to please wait for a while. Sir is joining. So sir will be joining within a few minutes. So participants are requested kindly post your questions during the session and after the completion of the session, so that you can learn more things, so that they can explain you in the same way, so that you can understand and you can apply those technology in your particular domain. So sir will be joining within a few minutes. So please wait for a few minutes only. Those who are interested in any publication, you can submit your paper to us and also to the official website of our society. You can share your paper with Dr. Jitendra Mehta. And if you are interested in any book publication, any paper publication, any patent publication or patent registration and grant also, in case of patent, your patent will be published and granted. In case of your design will be registered. So those who are interested in design registration, you can submit your request to Dr. Jitendra Mehta. And also we are also providing the technical assistance for patent filing. Even we are providing the assistance in the drafting, content writing, till filing and publication. So for that particular purpose, that particular work, you can ask for an assistance to Dr. Jitendra Mehta. He will be providing you
Dr.
same type
Shamsher,
of
audible,
same
sir?
type of assistance.
Dr. Shamsher.
So we are also providing several other type of assistance in conference, workshops, seminars. So you can also ask for association with you, and in the same domain for your academic credibility, you can
Dr.
ask
Nisha,
for the
I
help.
think he is
Sir.
joined now. He is
Okay,
joined.
sir. Okay, sir. So we will discuss the same things later. So I think the guest speaker has already joined for technical session 2. So now I would like to request Dr. S. Kumar Das, Associate Professor, Department of Veterinary and Animal Husbandry Extension, College of Veterinary Science and Animal Husbandry, Central Agriculture University, Imphal, Nagaland, to kindly deliver his lecture note. So kindly deliver your keynote lecture for technical session 2.
Okay, thank you. Respected participants, I would like to deliver my lecture on entrepreneurship in piggery development. PPT can be okay.
No issue, sir. Sir will be sharing your.
Already shared, sir, PPT.
Yes, shared.
You
But
just send.
Yes, sir. It's
You just
visible
inform
on.
me like next.
Sir,
okay, next PPT.
Yeah.
Entrepreneurship in piggery development. Next PPT. In 2025, economics, Nobel Prize in Economics was awarded to 3 persons for having identified the prerequisites for sustained growth through technological progress and for the theory of sustained growth through creative destruction. Next, please. According to Joseph Schumpeter, entrepreneur is a dynamic person who borrows capital. No, I think before that one slide missed. So in 2025, Nobel Prize was awarded in economics. Nobel Prize was awarded to 3 persons for creative destruction, actually entrepreneurship. So entrepreneurship based on creative destruction. Now, this creative destruction, this phrase was coined by Joseph Schumpeter in his book, The Theory of Economic Development. This creative destruction is described by him as the, no, before that, creative destruction described by Schumpeter, Joseph Schumpeter, as the process of carrying out new combinations. Carrying out new combinations of what? Factors of production. The factors of production, 4 types: land, labor, capital, organization. So, entrepreneurship, according to Schumpeter, means carrying out of new combinations of factors of production. Land, labor already there, natural, original factors. And capital and organization, that is enterprise, that is man-made. So these things are already there, but how these things are employed, used in innovative way, that was the issue for Schumpeter. So entrepreneur, according to him, entrepreneur is a dynamic person who borrows capital to combine factors of production in an innovative way to produce and market new goods to earn profit. Now, this carrying out of new combinations, according to Schumpeter, this carrying out of new combinations, there are 5 ways of carrying out of new combinations. Next slide. 5 ways. That is innovative entrepreneurship. 5 ways. One is introduction of a new good or a new quality of a good, introduction of a new method of production, opening of a new market, new source of raw materials or half manufactured goods, and carrying out of the new enterprise. These are the 5 ways of carrying out new combinations of factors of production, according to Schumpeter. Next slide. Schumpeter used creative destruction to describe the dynamic and innovative nature of entrepreneurship. Entrepreneur disturbs the stationary circular flow of economy, introducing innovation. Entrepreneurship represents disequilibrium, breaking the routine circular flow. He used static to express stationary circular flow and dynamic to express economic development through entrepreneurship. Next slide. Thus, Schumpeterian entrepreneurship based on creative destruction is largely absent in the developing countries, where entrepreneurship is confined mainly to borrow and adopt the innovation of the developed countries. So the point is that actually entrepreneurship, startups, these things are nowadays become very much popular, and in every sector, every aspect of life, these things are being applied. So livestock sector also no exception. So but that entrepreneur, this is not actually entrepreneurship, this is only the business actually, but whatever it may be. So now we will come to the point, actual thing, that is entrepreneurship development, entrepreneurship in piggery development. Next slide. Okay, I think now the PPT ends here actually. Now the whatever I want to express, that is in PDF form, PDF file. PDF, is it possible to share?
Yes.
Okay. Okay, thank you. So actually that part I wanted to share with the participants, because entrepreneurship, that entrepreneurship is not true entrepreneurship, but still it is business. So there is a difference between business and entrepreneurship. Anyway, now entrepreneurship in piggery development. So whenever a person wants to start an enterprise, establish an enterprise, he has to do it in a befitting manner. He has to do it systematically, keeping the records of everything. Then only it will be successful. Otherwise, livestock production as a way of living, that is different thing. So the livestock production, the economics of livestock production, that is the in entrepreneurship we will discuss this thing. So participants, I as a veterinary officer, when a farmer wants to establish an enterprise, they come to you, but not always, for taking borrowing bank loan under different rural development schemes. And that time a entrepreneurial project report is to be prepared, where veterinary officers uh, veterinary officer help is required, his signature is required. Without this it is not possible. Now you know this thing, I hope that you know this thing. But now I will share it with you. Now you think and you compare it, what you do and what I am telling, whether it is whether I am correct or not. You are doing actually practically. You are doing. I will talk share theoretically. So let us begin. So first of all, we have to make some assumptions that a farmer wants to establish a piggery enterprise. So this is assumption. In this way, so many things, that what type of piggery enterprise, piggery, what enterprise, that piglets will be sold, or pig after maturity, market age, it will be sold for meat purpose. So many things are possible. Here one example is given: to prepare an entrepreneurial project report for a poor farmer who wants to establish and run a small hog farm, obtaining bank loan under the government scheme. So many assumptions we have to make. Without these assumptions, it is not possible. Assumptions must be economically, technically, and technically feasible and realistic. For example, if feed price is in the market, if feed price is 30 rupees per kg, then that information must be written properly. Then only we will calculate. So there are so many assumptions, complete assumptions written here. Next slide, please. I will just share with you that this is the way to make, means complete way to make entrepreneurial project report. This may be, there may be discrepancies between what you do and what I am telling, but it will be good if I know from you that what you actually do and what I am telling. So is there any difference or something I want to learn from you? So that is the main purpose actually. So first of all, based on the assumptions, we have to calculate income and expenditure. So there actually this thing is nowhere written in such way that a person can learn it, understand it and learn it. Everywhere I have found this thing written, only calculation, some figure, assumptions of course there, but it is difficult to learn. I faced great difficulty in learning this thing. So first thing is that capital investment, and main problem is that it is sometime it is written capital investment, sometime capital expenditure, something is written not concrete, confusing. So it is capital investment, one-time investment. What things? Cost of animal, cost of shed, and cost of equipment. These are the three things under capital investment. And project for 5 years. Here it will be shown for six years. Actually, first year production income will be no income. That's why 6th year. So this is only for bank loan, obtaining bank loan. Bankers will be satisfied that yes, this enterprise is viable, so we can fund it. That is the purpose. Otherwise, the farmer will continue the project, continue his business. So that is the thing. Now up to 6th year, we have to make the project, and then also book value, etc., will be written. So capital investment, cost of animal, cost of shed, and cost of equipment. What I have done here below, next slide, we can see that, and not now, we'll see that how it is calculated, that is also written. Then total capital investment, then loan amount, after that fixed cost, variable cost. Fixed cost includes interest on capital, depreciation, insurance. And variable cost includes cost of veterinary services, electricity, miscellaneous cost, cost of feed. Cost of feed includes cost of feed for boar, sow, fattener, piglets. Then total cost of feed, then total variable cost. And next slide. Then one calculation variable cost per animal, because this project, entrepreneurial project, is for hog farm, means animal will be sold at the age, marketing age, for meat purpose. So cost per animal must be calculated. Then gross expenditure, that is fixed plus variable cost. Then gross income, sale of animal, sale of empty feed bags. After that, net income per annum, net income per month. Now thing is that what I have done here, this is the every calculation is explained below, how the things are calculated. So anybody, if carefully go through this slowly, carefully, then he can understand. And that is the main purpose of doing this thing. Of course, all of you participants know this thing and do this thing day-to-day life, practically. But perhaps some of you may have some doubt also. So that's why I thought that this will be the um, this can be the good way to express that entrepreneurship in piggery development. That's why I am sharing this thing with you. Next slide. Next. Next. Next slide, please. Cash flow. No. Before that. Before. Yes, okay. So first one, income and expenditure, that was done. Now this is production chart. Everywhere in NABARD material, everywhere I searched it, I found everything written, the calculation, but how this thing calculated, that is not written anywhere, because we should not expect also that those material, everything will be written in this way. But this thing I have prepared for learning purpose. That's why everything is written here. So first of all, we have to know we have to do this. This production chart is essential. Without this, we cannot calculate. First of all, opening stock every year up to 6th year, male, female, young, adult, male, female. We have to calculate as per the assumption. Opening stock, number of animals born, number of animals died. In assumption, we have assumed percentage of mortality. Then number of animals alive, number of fatteners sold, number of breeding stock sold, number of animals retained, closing stock every year. Then also calculation is shown below. And then also every year some means we have to keep replacement stock also. The economics of this enterprise means every cost must be calculated, and so every cost must be calculated. No cost will be left. No cost will be left uncalculated. And another point is that no cost will be repeated. So this is the thing. Economics is not the list of grocery. Economics means this thing only. Even opportunity cost also. If the farmer invest instead of doing this thing, if he employs himself in other business, other purpose, if he earns more than this, then what is the meaning? What is the point of doing this business? One person must do this business when he will find that he is nowhere suitable, he cannot do anything but only this thing. So in this way, opportunity cost also we must calculate. Next slide. And then loan repayment schedule, first, second, up to 6th year, principal, interest, repayment, outstanding loan, net operating income. Net operating income is what? Everything is explained. That principal, interest in the form of superscript. Some symbol is given and below it is explained. Next slide. Then cash flow statement. Operating expenditure, that is cash outflow. It includes insurance, cost of veterinary, electricity, miscellaneous cost and feed cost. These are the 1, 2, 3, 4, 5 items, operating expenditure. Then gross income, cash inflow, that is income, cash inflow, that is animals sold and empty feedback sold. Then net operating income per month, debt burden, then DSCR, debt service coverage ratio. Everything is explained and calculated. This is one example. Then next slide. And another statement required, that is economic viability statement, where expenditure, gross income, net income, discount factor, discounted cost, discounted benefit, net present value, NPV, benefit cost ratio, IRR, everything is shown. And now point is that discount factor. Actually, my purpose is to learn from you. Means something at least, that no, whatever you told, that is very different. Something I want to know. So discount factor 15% means it is assumed that if I keep this money capital required for this doing this small scale business minimum for a poor farmer, if instead of investing that money in this business, if the farmer keeps this money, saves this money in the bank, he will get some interest. So that interest, he will get money, income. Now this business must give him more income than interest, bank interest. Then only it will be meaningful. Otherwise, what is the meaning? So that's why it is assumed that bank will give loan, soft loan means under different development scheme. So minimum means rate of interest will be less. it is assumed that 10%, but in real life may be different anyway. So 10% interest. And now if the farmer has to earn profit, he must earn more than 10% interest. Otherwise, what is the meaning? That's why it is assumed that 15% interest rate. There is discount factor and discounted cost, discounted benefit calculated in this way. All these things can be done in Excel sheet, that is spreadsheet, easily, without doing manually. And that means if we put the assumption and then accordingly with the formula, we can do all these things very easily. Particularly IRR, internal rate of return. Calculation of IRR manually, I don't know. Theoretically, I know, but it is nearly impossible type. But in spreadsheet, Excel sheet, it is very easy. IRR. So IRR, DSCR, debt, BCR, benefit cost ratio must be more than 1. If it is less than 1, that is project is not viable. So in this way, this thing, economic viability statement. Now another point, another aspect bank wants to know, that is break-even point. The point where no profit, no loss situation. So that break-even point calculation in terms of output in number or unit and output in rupees. Both ways it is to be shown. And then only we can recommend, means conclude that based on the techno-economic assumptions and the economics and project appraisal done above, it is recommended that the project is technically feasible and economically viable, and the project may be accepted and approved for funding. Okay, with this I end my presentation. Thank you for listening.
So thank you so much, sir. So participants are also requested to kindly post their question in the chat box.
Thank you, Dr. Shamsher. Nice presentation.
May I request to all the participants to kindly post their questions in the chat box. So we'll be waiting for 2 minutes only, so that if there is any query, so they can drop in the chat box.
I think no question.
If there is no query, so thank you so much, sir, for enlightening us for such an informative session. Thank you. So, on behalf of the organizing committee, I would like to express our sincere gratitude to all the dignitaries, our speakers, and participants. Thank you everyone for your active participation in today's enriching session. With this, we conclude the day 5 of 10 days International Training Come Workshop on advances and innovation in veterinary science for viable livestock practices. We look forward to your presence tomorrow for another enriching session with our distinguished speakers. Thank you once again and Jai Hind to all.
Thank you.